Hello, our names are Charles, Via, and Tim.
We are seventh grades from DIS.
We are here to talk about DNA connection.
DNA is quite interesting. It stands for deoxyribonucleic acid.
Chromosomes contain DNA in them.
DNA molecules are made up of 4 different nitrogen bases, and they're adenine, thymine, guanine, and cytosine.
A gene is a section of DNA molecule that contains the information to code for a specific protein.
Also, the bases in a gene are arranged in a specific order. A single gene in a chromosome may contain anywhere from several hundred to a million or more of these bases. All of the genes are on a specific place in the chromosome.
A gene contains the code that determines the structure of a protein.
The structure of the specific proteins depend on the order of the nitrogen bases along a gene. Proteins are long chains of amino acids.
3 DNA bases code for one amino acid. Remember that the base codes are adenine, thymine, guanine, and cytosine.
Protein synthesis is the production of proteins. This is when the cell uses information from a gene on a chromosome to produce specific protein, and protein synthesis takes place on the ribosomes in the cell's cytoplasm.
The genetic code from the DNA is carried into the cytoplasm using RNA. RNA stands for ribonucleic acid. The RNA that carries the genetic code is called the messenger RNA.
RNA is different from DNA. RNA has only one strand instead of 2. Also, RNA contains a different sugar molecule from DNA.
RNA contain adenine, guanine, and cytosine like DNA, but instead of thymine, they have uracil.
The messenger RNA copies the coded message from the DNA and carries the message to the ribosome. The transfer RNA carries amino acids to the ribosome and adds them to the protein.
The first step of protein synthesis is when the DNA splits and the genetic information in the DNA is transferred to the messenger RNA strand.
Then, the RNA goes and attaches to the ribosome. Now the transfer RNAs come and read 3 bases at a time and links the amino acid that it was carrying as the ribosome moves along the messenger RNA strand.
All the amino acids link up until the ribosome reaches a 3-letter code that acts as a stop sign. This is the full process of protein synthesis.
If a mistake occurred in one gene of a chromosome, it is called a mutation. These mistakes can cause a cell to make a wrong protein in protein synthesis, so the phenotype of the organism might be different from normal.
Some of the examples are having 6 fingers or toes, having 2 heads, or even having blue eyes.
There are different types of mutations. There can be a substitute for another base pair, or a removed base pair, or an added base pair.
Other mutations might occur if the chromosomes didn't separate correctly in meiosis. This will leave the cell with too many or too few chromosomes. The cell could also end up having extra segments of chromosomes.
Mutation can be a source of genetic variety because it changes an organism. Some mutations are helpful, some are harmful, and some are not helpful or harmful.
For example, if an animal is white, this can be harmful because it can be spotted. If something is immune to some kind of illness, then it is helpful.
So to recap, protein synthesis is the production of proteins, and proteins are made by the ribosome and RNA. Also, mutations are any changes in a gene or chromosome. This was Charles, Via, and Tim from the DIS 7th grade. We have just taught you about the DNA connection.